I hope to convince you at the end of this talk that it is time to place genomics at the forefront of dementia care. And I'll start with this poster of a very young-looking Gough Whitlam and his phrase, "It's time," which was such a successful political campaign that propelled Gough Whitlam to be our 21st Prime Minister back in 1972. Now, for you who actually are old enough to remember that moment that occurred 48 years ago, what it means actually is that you actually reached an age when you should start worrying about your risk of dementia, because dementia risk increases substantially after the age of 65. For this talk, I'll be talking about frontotemporal dementias, which are the most common cause of young onset dementias. And it's also interesting because familial forms in this kind of dementia is actually surprisingly common. More than 30% of the family of patients actually have some form of family history where 2 or more people are reported to have some form of disease as well. And as I said before, what I hope to convince you is actually the need to integrate both basic and clinical science. So for a patient who undertakes genomic screening, the patient fronts up to a clinician, blood is collected, and the DNA is sent to a sequencing facility. Other laboratories may be contacted to do more phenotyping. Results are fed back to the clinic, and that give rise to hopefully to good patient care. What we should realize actually is that basic research impact positively on every step of the patient's journey. For example, from our work, when we discover a new disease gene, that gene can now be offered as part of the testing panel for the patient. Functional genomics, which is the science of understanding how a genetic variant gives rise to disease, can also be improved by increasing the throughput of the variants that have been increased. All the types of cellular assays that we can do can be developed to answer that question as well. Ideally, clever bioinformaticians then integrate all this information to basically help us understand underlying disease mechanisms, and in doing so, help generate better biomarkers and, most importantly, novel therapeutics. So I'll start my example with this patient and a gene called C9orf72. So C9orf72 is actually the most common genetic cause of frontotemporal dementias and motor neuron disease. The mutation is actually a hexanucleotide repeat expansions in the intron of the gene. And that gives rise to a very distinct pattern of neuropathology. Now, the repeat expansion can be detected using repeat prime PCR. And the result is visualized in this gel, where you see this very characteristic sawtooth pattern, where each individual peak corresponds to a single increase in repeats. The original paper suggested that if a patient has more than 30 repeats, then it is classified as pathogenic. This does bring us to our first case study. So this is the 40-year-old patient with motor neuron disease. As you can see here, there's not much indication of family history since both parents are in the late 60s without any apparent disease. And yet, because we know that sporadic people can actually have the repeat expansion itself, she was screened for the C9orf repeat expansion. As you can see here, the patient actually had about 28 repeats, and that was actually the cause of her diagnostic odyssey. So what happened was that in the original lab, the research lab, because of technical difficulties, they were only able to say that that patient had more than 15 repeats. And so the results was inconclusive. The patient was then referred to another lab, research lab, and they were able to show that that patient had 28 repeats. Now, using the original cutoff of 30 repeats, that patient was told she was negative for the repeat expansion. Because it was inconsistent between the 2 reports, the genetic counselor reached out to the first NATO-accredited laboratory, and they said that patient has more than 23 repeats. and verbally reported that the patient had a pathogenic mutation. Since then, another national accredited laboratory was contacted, and their results agreed with the second research laboratory in that the patient had only 28 repeats. The difference is that 28 repeats now is considered to be intermediate, which has got some clinical significance, but not much is known about it. So that poor patient's diagnostic odyssey went from inconclusive to negative, back to positive, and then finally to intermediate. And what it tells us that there's much clearer need for the laboratories and clinic to standardize testing protocols and the way that we interpret the genetic data. The second case study it's a much brighter note, and actually this is a family that was identified by Professor Clement Lloyd back in 2013 for his PhD thesis. So this is the proband actually had some family history. So the proband's mother became forgetful at age 72, and she actually passed away at age 75 from dementia-related condition. Her cousin from the maternal side also had frontal temporal dementias, but at a very early age of 52. The proband herself became apathetic at age 45, and then she rapidly degenerated by age 51. At this stage, she was unable to dress for herself, she lost the ability to speak, and she became doubly incontinent. And Clement diagnosed her with frontal temporal dementias, and genetic screening was requested for the ghrelin gene, which is the second most common cause of frontal temporal dementias. So this is, the mutation that was found was actually what they call a loss of function mutation, which introduces a premature stop codon at the 300 amino acid of the ghrelin gene. Here's an example of the few remaining wet lab data that Clement Lloyd produced back for his thesis. And he showed using a Western blot that that mutation basically stopped full-length protein to be produced. Now, since the discovery of the gene, we know a lot more about its function. We know that the ghrelin protein plays a key role in inflammation by its interaction with the microglia. But more critically, we know how that protein is degraded in a cell. So the protein is degraded in the lysosome, and the protein sortilin plays a part in it. And this information led directly to a new form of therapeutics in which a monoclonal antibody raised against its sortilin-1 is infused into the brains of mutation carriers. And by reducing sortilin-1, we reduce the amount of degradation of the ghrelin protein. And in doing so, we hope to raise the levels of ghrelin protein back to normal levels. And so the good news is that the sister of the proband has been confirmed with to have a mutation, and she's now able to be recruited into the clinical trial, the phase 3 clinical trial. The last part of my talk, I'll move slightly away from dementias, but talk about a neurological condition called chemotherapy-induced peripheral neuropathy. And this anecdote actually highlights why it's actually important to listen to the patient. So chemotherapy-induced peripheral neuropathy, or CIPN, afflicts over 30% of cancer survivors. The cancer patients can live with these painful conditions for a very long time, and there are actually no truly efficacious therapeutics. So there is one compound called duloxetine, which has got minimal effects. And that actual condition actually can limit the effective use of a number of chemotherapeutics because the patient cannot tolerate the neuropathy pain. And this is just a diagram schematic to show the main symptoms occur in the hands and feet, and that the main forms of chemotherapeutic drugs like the taxanes can impact on the microtubules of the nerve cells. So the study was actually led by Professor Susanna Parks from Brain Mind Center, as you all know. She looked at the ICON-7 data set, which comprised 150 1,528 ovarian cancer patients who were treated with a combination of carboplatin and or paclitaxel. What she was able to take from the data set is that there were two different measures of neuropathy, a patient-reported outcome versus the clinical reported outcome. So the patient outcome, or ERTC, basically asks the patients the symptoms. Do you have tinglings in your hand and feet? Do you have shooting or burning pains in your toes or feet? Versus a more clinical reported outcome where they measure a more objective loss of deep tendon reflexes, sensory loss, or paresthesia, which is basically pins and needles. And what she found is actually that the patient-reported outcome was more sensitive measure where the patients were able to show that the high incidence of neuropathy if you use the patient-reported outcome. What I was able to show is that the genetic loci, which predicts whether you develop the neuropathy, differ depending on the outcome measure as well. So to sort of try and address these issues in a more systematic manner, we conducted a genome-wide sequence association study, as you've heard from what. And what basically involved is we take a large cohort of cancer patients and divide them into either people with the neuropathy or without neuropathy. And we measure the genotypes of over 800,000 common variants. And each variant is then examined for the association with the, of the neuropathy. So we, the final cohort was actually 234 breast cancer patients that were treated with paclitaxel. 225 patients were successfully genotyped using the Illumina Infinite genome screen array chip. And after QC, we ended up with 206 cases that we took to the final GWAS and pathway analysis. But the critical thing to note is this QQ plot, which plots the observed p-value versus the expected p-value. And the critical take-home message is that the patient-reported outcome, the EORTC, was basically much more sensitive in picking up the neuropathy for the majority of the genetic variants we looked at compared to the clinical reported outcome. And in fact, 4 genetic loci with p-values of 10 to the minus 8 were identified, and this actually exceeded genome-wide significance for genome-wide array association studies. Now, to dig deeper into the data, a simple thing that we can do is actually to assign the gene, corresponding gene with the, from the genetic variants and see which genes are consistently impacted by predict the neuropathy. And so here's just basically, we actually looked at 4 different measures of CIPN. What we found was that the most common pathway that was interrupted by the paclitaxel treatment was the G protein coupled receptor pathway, which is something you've heard from Professor from this morning's talk. The most critical thing that we note was that 2 genes, which correspond to the sphingosine 1 phosphate receptors, S1PR1 and S1PR4, was actually replicated in a very recent meta-analysis of multiple GWAS studies by Chuertel back in 2020. And for people who work with multiple sclerosis, your attention probably noticed that of the compound fingolimod, which actually used to treat multiple sclerosis and actually impact on the S1PR1 pathway, has actually been approved for phase 1 clinical trial for the neuropathy in 2020. And finally, what we'd like to highlight actually is the advantage of working at the Brain Mind Center, in that we have access to areas of expertise that will help us do our research. So for example, the fact that we're interested in the S1PR receptors, we can reach out to Associate Professor Anthony Don from the Center Institute, who just so happens to work on this particular pathways, and he was able to provide molecules for us to test. Another thing to note, gene that's impacted is actually the Gabanergic receptors. And and just by walking across to the next building, I was able to contact Professor Mary Collins. And her research interest is actually looking at the Gabanergic receptors and precision medicine for epilepsy. But again, she was able to suggest a drug that we can use for our studies, baclofen. And the final thing we can say is actually some of the novel genes, like the adrenergic B2 receptor, has just recently been found to impact on pain reception through the epinephrine pathway. And we hope to reach out to people like Professor Mark Cassio to design small molecules that can actually be impacted on this as well. And so on this note, I will finish my talk and I'll be happy to take any questions.
Okay, thank you, John. That was fascinating. We have a moment or two if people would like to pop questions in the Q&A. Please don't be shy. In the meantime, I have a question. To me, it seems really surprising that there would be such a divergence between what the patient-reported outcomes and the objective sort of clinical measures tell you in relation to the peripheral neuropathy. Was that really unexpected too? And do you have any ideas of what sort of underlying that?
I actually don't know. I think people who work in the neuropathy field probably be able to give you a much better answer to that. But what strikes me is actually, in terms of the dementia field, we're actually looking at that as well, because I think Sharon Naismith is actually looking at the impact of subjective cognitive impairment as a sensitive measure. And she found that that is actually a very good way to predict subsequent clinical loss. So I think actually just, a person knows his or her body better than anybody else. And you can, a perceptive person would be able to recall or notice subtle changes much before anything else that the doctor can pick up at a later stage. So I think that's the basic. I think we do know our bodies.
Yeah, it's really interesting. It might be something that sort of scales with the severity of the neuropathy, that, at sort of milder levels, that the patient report might be more sensitive. But
that's
then
right.
as far as you might see them, as it's more severe, the two methods might converge. So
yeah, you can do correlation analysis to show that they are correlated in terms of the ability to predict. But if you do a GWAS, the patient-reported outcome is just much more sensitive at picking up the genetic loci.
Yeah, that's very cool. Thank you.